Hey guys, so I'm going to go over a couple of the daily brainies this week. As you know, these provide a lot of practice for kinds of questions that you're going to be seeing on the exam, on the AP exam. But you also are going to see these on the DCAs. So I'm going to be from here on out probably going over some of them a little bit more because I think a lot of you are making simple mistakes that we could fix just because of how you're looking at the questions and how you're eliminating them. Okay, so for example, for this one here, it says the antibiotics interfere with prokaryotic cell functions. Streptomycin is an antibiotic that affects the small ribosomal subunit in prokaryotes. Specifically, streptomycin interferes with the proper binding of tRNA to mRNA in prokaryotic ribosomes. Which of the following best predicts the most direct effect of exposing prokaryotic cells to streptomycin? Okay, so basically what's happening here is it gives you information about this antibiotic here, and it is talking about how it affects the small ribosomal subunit, and it's affecting the binding of tRNA. So you have to think to yourself, okay, what are those things involved in, first of all, okay? And we know that after transcription, the next part of the phase is translation, and that is where the ribosomes, the small and the large unit, come together on the mRNA and bring tRNAs carrying amino acids to the site to build the polypeptide. Okay, so now you have to look at the questions. This first one was one that a lot of you picked. It says that amino acid synthesis will be inhibited. And here's why this is not the right answer. Because the term amino acid synthesis means that it's a process that is making amino acids. Well, think back to translation. We're not making amino acids. We're just using amino acids that are being brought to the site with tRNAs to make, what are we making? We're making a polypeptide. We're making the protein. So the reason this one is incorrect is because we're not making amino acids in this step. We're just using already made amino acids that are made somewhere else. We're using them to build a polypeptide. Okay, this one is incorrect because it says that no mRNA will be transcribed. Well, these two things are not involved in transcription. And the same thing here, post-translational modifications. That is after the protein is released from the ribosome and it goes through its modification and folds up the way it needs to fold up. Well, the ribosome has nothing to do with that anymore because it's been released from it. Okay, so those three are incorrect. Making the polypeptides will be inhibited. Okay, I hope that helps. All right, so here is another one. I believe this is one you guys had on Wednesday. This gives us some information about an antibody. So it says nuclear pores regulate the passage of substance into and out of the nucleus. Antibodies such as the MAB414 have been used to inhibit the movement of substances through the nuclear pores of rat liver cells. Scientists cultured rat liver cells, eukaryotic, and bacterial cells, prokaryotic, in separate dishes with radioactively labeled amino acids. A specific gene in both cell types was engineered to synthesize identical polypeptide chains, and translation of the gene was measured. The procedure was repeated with the MAB414, the inhibitor, added to each of the two cultures, and translation was monitored again. Which of the following sets of graphs best summarizes the results of this experiment? Now, this gives you basically a whole bunch of complicated information. Really, this question is basically comes down to one simple fact. What's the difference between transcription and translation in prokaryotes and eukaryotes? Okay, so we have these lines. The solid line is the eukaryote, the dotted line is the prokaryote. And this first column here, these first options are with no inhibitor. Okay, so they just were allowed to do their thing, and you can see that transcription and or translation worked in both cultures, no problem in both prokaryotes and eukaryotes. Okay, now this second one here, this is the experimental. This is the one where they added the inhibitor that is going to prevent things from going in and out of nuclear pores. Now, all you need to know about this is to know that, okay, Well, a prokaryotic cell does not have a nuclear pore. It doesn't, things don't leave the nucleus to get translated like they do in the eukaryotic cell. Okay? However, in the eukaryotic cell, if that transcribed mRNA cannot leave the nucleus, that means that translation isn't going to happen and it's going to be hindered. Okay? So, in that case, we would expect to see that the prokaryotic cell would continue as normal, right? These dotted lines are identical because the prokaryotic cell, it doesn't have a nuclear pore, so nothing is being inhibited, so it shouldn't change it at all, right? And then here, you see the eukaryotic line being stagnant. Nothing's happening. There's no translation. Well, we would expect to see that if the nucleus if things are not leaving the nucleus. Okay, so this is our correct answer here. And I just want to point out too that even just knowing that prokaryotes don't have a nucleus, you should have been able to eliminate this one, right? Because we know it doesn't have a nucleus, so it should be the same. And that one would be eliminated as well, because if it doesn't have a nucleus, nothing's being inhibited, nothing's being changed. So being able to eliminate some answers are really important as well. And then this one is just kind of one to throw you off because your options are from between here and here. And it's really just measuring translation. And either things get out of the pore and they get translated, so it would be a straight line, or they don't, and it's a flat line like this. So in this case, this is your answer. And again, you really have to kind of look at these questions and boil them down. It's like, what is it that it's wanting us to know? And in this case, you just needed to know that a prokaryote doesn't have a nucleus and then be able to reason through the examples that they give you. Okay, so this one was Thursday's Daily Brainy, and this one gives you a lot of information about lactose digestion in E. coli. And this is what you did the simulation on and did some notes on. This is the lac operon. So basically it says that lactose digestion in E. coli begins with the hydrolysis of the enzyme beta-galactase, and the gene encoding beta-galactase, LAC-Z, is part of a coordinately regulated operon containing other genes required for lactose utilization. Which of the following features correctly depicts the interactions of the lac operon when lactose is not being utilized? Okay, and then the legend gives you the different shapes and what they are. Okay, so obviously we have a lac operon, we have our promoter, our operon, or our operator, we have a repressor, and we have our genes. So we have everything that we need for an operon. We know that RNA polymerase binds to the promoter, and if the operon operator is here, or the repressor is bound to the operator, it's going to prevent the translation or the transcription of all of these genes downstream of it. Okay, so here, this is where these get tricky, because you look at one image, right, when you're studying it, you see an example of it, and then they switch it on you, and now it looks a little bit different. So you have to be able to really look at keys and things like that to make sure you really are understanding what's happening here. Okay, so this one that gives you this little shape, this almond shape, is a polymerase. This is your repressor protein, and this is your lactose shape. Now, this is different than the simulation we did, but as long as you have the key, you should be able to kind of work your way through the examples and figure it out. Okay, so now, what is the situation? There is the lactose is not being utilized, meaning this E. coli is in someone's stomach and they didn't have any dairy today, so they are only working on digesting carbohydrates or proteins that day, but there's no lactose around, so this process doesn't need to happen. So that E. coli now is digesting something else, and so this process doesn't have to happen. Okay, so what you have to figure out then is, what would this operon look like? Okay, now in these two, what do we see? We see this lactose, right? Now, would we see lactose if there is no lactose, we wouldn't. Okay, so there would be no lactose here. So these two you should be able to eliminate, because if we're not using lactose, there's no lactose, and there's lactose in these pictures. So that leaves us these last two. And so we have to figure out what would happen between the two. Well, if lactose is not present, then this repressor protein we know would be bound to the operator. Well, this one is the only one where the repressor is repressing these genes. And so this is your option. This is your answer. Here, first of all, this RNA polymerase is bound to the operator and not the promoter, where it would be, bound, unless it was in mid-process, in which case, again, we wouldn't want this process to proceed if no lactose is present, right? Because this is going to produce proteins that are going to break down lactose. And if there's no lactose present, then this process is a waste of time and energy. So really the only option here is this one, where this repressor is bound to the operator, so that even if an RNA polymerase binds to it, it's not going to transcribe these genes. And it will remain there until that person whose gut these E. coli are living in eats something with lactose in it, and then that lactose will bind here, removing this, and then this process will happen. Okay? Okay, and finally, Friday's Daily Brainy. This is again another image of the lac operon. And in this one, interestingly enough, it doesn't show the promoter, but know that there's a promoter here where obviously this one must be the promoter, if that's where polymerase is bound, but it doesn't show it for whatever reason, which is another thing that gets kind of confusing when you're trying to answer questions like this. Did they leave it out because it was unnecessary? You never can really tell, so you got to kind of fill in as much information as you can. So basically it shows you the LAG, Z, Y, and A genes. It shows you the repressor, and it shows this one in a case is called allolactose. It's an isomer of lactose, which would be present if lactose was present. It's basically a mirror image of lactose, and it will bind to the repressor the same way. So it says that it binds to it. The cap site won't let this process happen, won't let RNA polymerase bind if glucose is present. So it needs lactose and allolactose in order for these genes to be transcribed so that they can digest the lactose. So basically this image here it's asking you for a scientific claim that is consistent with the information provided. And I know these are your guys' favorite questions, because you're basically trying to figure out, like, more than one of these may be true, but you got to find the one that is making a claim that's consistent with the image. What is the image showing us? Okay, so the first thing says that the presence of excess lactose blocks the functioning of RNA polymerase in this operon. So if there's extra lactose, will it block the function of RNA polymerase? So you gotta kind of think back to what you know about the lac operon. We know that in the lac operon, the presence of lactose actually turns on the translation of those, or transcription of those genes, because if lactose is present, then we want to transcribe those genes so that the bacteria can digest lactose. So this one is we know that this is incorrect just based on our knowledge of the lac operon. This one says, when bound to the operator, the repressor protein prevents lactose metabolism in E. coli. And this one, again, we know this, right? Because we know a little bit about the lac operon. We know that when the repressor is bound to the operon, in this case, it prevents the transcription of these genes, and then they will not, and if those genes don't make the proteins that they need, then lactose will not be digested. Okay, so in this case, B is the correct answer. I just want to kind of go through the other answers here. The binding of the repressor protein to the operator enables E. coli to metabolize lactose. This is obviously incorrect. The repressor protein physically attached to the operator is what actually prevents, not enables, the initiation of transcription. So lactose is not metabolized in this situation. And then the last one, the allolactose, allolactose acts as an inducer that binds to the operator, allowing E. coli to metabolize lactose. It does allow E. coli to metabolize lactose, however, it binds to the repressor protein, not the operator sequence. Okay, it acts the same as lactose in this case by binding to the repressor. And this one we should be able to eliminate just by looking at the picture because it says that it binds to the operator and it clearly shows it bound to the repressor. Okay,